Dr. Banya, can we check how many delegates logged in?
Sir, अभी no, none of the delegates have logged in because attendees में 0 है.
Okay. So can you send a message in the group?
Okay, sir. I'll just share.
Are they finding some trouble in logging in?
Hello. Hello.
Yeah, hello. Yeah, I'm on. Hello, Dr. Kalita.
Yes, sir.
Yeah. I think the antivirus is doing some mischief here. I have just called our software person. But meantime, we can continue. Yeah.
Okay, sir.
Yeah.
So he will be working on this, I think, your IT person.
Huh?
I mean, in the meantime.
Yeah. He's coming.
Okay, sir. Because we want to have you live, actually. That is the reason. Okay, sir. Okay, no issue.
Yeah, no problem. I am live. Only thing is I am not seen. My camera is on. I think the computer, some settings, the antivirus, I think, is
okay, understood. Okay, sir. Okay.
I think so. I don't know. Anyway, let him come and sort it out.
All right, sir. All right. No issue. So we are waiting for the participants.
No issues. I'm online.
Dr. Banya, is there any update on that? How many people have joined, or because I'm not sure I can see it from here. Dr. Deepika?
Yes, sir, we are on it, sir.
I'm asking about the delegates,
Hello,
participants.
Hello, sir.
Yes.
I have talked to 2 delegates. Like they are already joined, but we are not able to see them, and even they don't know how many people are there. Like Dr. Anjali.
No, they are in.
So
they can see
hear
us.
Yes, sir. They can hear us as well.
Okay.
So
how many? Two of them.
The 2 to I have talked. So I think this Balkarji, are you still there?
Yes, ma'am. Yes.
Kindly see to it. How will we know how many people are there, because people have joined in here?
Ma'am, total John sir हैं, सुनीता, दीपिका चौधरी, वानिया जी.
Sir, ये तो panelist है ना, delegate who have
delegate. Okay.
Yes,
okay. अभी confirm करके बता रहा हूँ. अभी.
Yes, sir.
Because we have the presenter as well as chairman is already there. So just 2 minutes left. We can start actually if the delegates are there.
Yes, sir. Delegates are there, just that we are not able to see them. These IT people, they'll help us now.
Okay. So in 1 minute we will start, and if the IT guy can update us about presence of delegates, will be comforting for us. We'll feel we can't see it from here. I think that thing, there is a issue we need to discuss later on.
Dr. Kalita, you can see me?
Yeah, absolutely. You are there.
Yes, sir.
I am trying to contact the IT guy. If he can update us, we want to start actually, because it's time now. Dr. Banya, no information as yet?
No, sir. We have informed the technical team, but no information from them. But like Neela ma'am, Dr. Ranjana, we have called a few people. They've already joined. It's just that we are not able
But
to see.
they can't see it, no?
Just
sir,
leave
they can
leave
see
and
it.
restart.
So you are visible on like other screen. Like
Hello,
delegates
am I
can see.
visible?
Yes, sir. Very much.
Hello?
So you are visible, sir. Kalita sir is visible.
No. John Jude, is it?
No, sir. We can hear your voice. We cannot see you. Sir.
Okay, I'm just leaving.
Ah,
And rejoin. I think.
okay, sir.
Yeah, right. Thank you.
सर स्टार्ट कर सकते हैं अभी.
Okay. I got a pop-up message which says that they want to unmute me. I'm already unmuted. Anyway, I'm talking. So, brief introduction about our speaker and the chairperson, because chairperson will be back very soon. I will start from the speaker. So our speaker today is Dr. KG Sunitha. You can see her. She is a very well-known name actually in the field of this IFA and diagnostic and research area of immunofluorescence and autoimmune diseases. She is basically, background of her is she's a training head of and group product manager of the CPC Diagnostic Private Limited, and she's into the marketing and planning division. Her research area is very vast actually. It includes biochemistry, cell biology, immunology, molecular biology. See into all these fields, enzymes and intermediary metabolism, very difficult area, biophysical. So just a minute. Zidane workshop शुरू हो गया है. Okay. Sorry. Workshop शुरू हो गया है. It's okay. All right. So achievement, she has actually won a lot of awards. I'm told that she has won the Chief Minister's Endowment Award for her master's degree, then Young Scientist Seminar Award. Then she has won this, very difficult for me, I'm not sure whether I can pronounce it correctly, Yalla Pragara Subbarao Memorial Award for a research paper. and she has won this CM Vijay Lakshmi Endowment Award for her masters, as well as, and most importantly for us, she has a lot of publications in autoimmune diseases, especially in autoimmune diseases besides others, and in important journals in this field. So she has won this first prize in paper presentation multiple times. She has conducted a lot of workshops, and she is a vastly experienced person in this field. Actually, they have been conducting workshop after workshop. I know about it, and she is a great resources for all the beginners, learners. And okay, thank you so much for the presentation. I mean, so then I go to Dr. John. Is he here? Has he joined? I can't see him actually.
He told that he's going back and will join, sir.
Sorry?
He said he will join back in few minutes.
So whenever he joins, I will talk about I will give his introduction. Is it okay? Or should I start, say about it now?
You can just check with him, sir, whether he's joining in a few minutes or what.
He is not joined yet.
Mhm.
He has not rejoined, I mean to say.
Sir, one confirmation I wanted to give,
Yeah, I can see him.
actually the speaker.
Welcome back, sir.
Yeah, thanks
We
for,
can see
for your patience.
now.
Yeah,
No,
yeah.
it's okay, sir. Actually, we have just started, and you were right. Back on time actually. And thank you so much, sir. So we were giving the introduction of our speaker as well as the chairperson. So our chairperson today is Dr. John Anthony Zoot-Procas. He is a professor of microbiology at CMC Vellore. We all know this one of the top institute in India, and he is also the quality manager of clinical microbiology there. And his research interests are in the field of development of new assays, especially rapid diagnostics in zoonosis and acute febrile diseases, I mean to say. And rickettsial infection is another area. And most importantly for us, he is very much into autoimmune diseases for a very long time. I'm told he is one of the oldest, not oldest, one of the most experienced person in the field of autoimmune diseases, is told by Dr. Sunita. Dr. Sunita told me yesterday. So he is into two areas like neurological and degenerative diseases, some of the most important applications of this IFA actually, and he is into it. Then his professional achievement, he was a visiting scientist in the University of Bethesda, USA. He worked in the Naval Medical Research Center of Silver Spring, MD, USA. And he is a reviewer of a lot of prestigious journals. I can see American Journal of Tropical Medicine and Hygiene, BMC Infectious Disease, PLOS, Science Report, and many of them. And he's a reviewer of SCRB also. And he is linked with many Indian journals of prestigious Indian journal, like Indian Journal of Medical Research, it's ICMR journal. He is linked with Indian Journal of Medical Microbiology. He has nearly 100 publications in the top journals, basically, national, internationals. So we are very lucky to have him here, and he is going to be a great asset for today for our learning process. And after this introduction, I will just briefly about the outline of this workshop. Immediately after, Dr. Sunita will take over the proceeding. She will speak. She will present her presentation. Once she finishes, Dr. John will actually take over. He will put his own feedback, his own input into the entire presentation. He might summarize, or he can add, and that will be followed by question and answers. And finally, we'll have the vote of thanks. So with these words, I want to hand over the proceeding to Dr. Sunita. So she can start her presentation now.
Yes.
Dr. Sunita, please.
Yeah. Thank you so much, sir, for the lovely introduction. It's a pleasure and privilege for me to join this workshop on IFT. I'll just share my screen. Yeah. I hope all of you can see my screen and my voice is audible.
Yes.
Yes. Yes.
Yeah.
Thank you so much. Yeah. So without much ado, I'll just start the presentation. In today's discussion, I'll be talking about the immunofluorescence, type of indirect immunofluorescence, type of immunofluorescence, direct and indirect method. We'll also discuss on autoimmune disease as a whole. We'll see what is the importance of doing IFT as far as ANA screening is concerned. We'll also discuss on the technique of indirect immunofluorescence and in relation with ANA and its types, and also on ANCA. So if you have any questions, we can discuss at the end of the session, as Dr. Kalita told. Yeah. Now, down the memory line, I would like to talk about Dr. Albert Kuhns, who is the pathologist and a physician who is called the father of immunofluorescence. So I just wanted to just share the screen of, talking about Albert Kuhns. So coming to immunofluorescence. So immunofluorescence basically depends on antigen-antibody reaction, where the antibodies are tagged, or you can say labeled, with a fluorescent dye. So in the case of indirect immunofluorescence, the dye that we use is FITC, fluorescein isothiocyanate. And the antigen-antibody complex is visualized using fluorescent microscope. So it's a simple antigen-antibody reaction. So here, as I was telling, we use a fluorochrome, which are dyes that absorb UV rays and emit visible light. So this process is called as fluorescence. Yeah. So coming to the types of immunofluorescence techniques, there are two types. All of us know about this, but anyway, for the benefit of people who don't know, I'm just telling about the basics. That is the direct immunofluorescence and the indirect immunofluorescence. So direct immunofluorescence, here in this, we take the antigen, which is from the patient sample. For example, in the case of bullous autoimmune disease, we take the skin biopsy. Then we add the fluorofluor, which is conjugated to the primary antibody. It's a single step process. Whereas in the case of indirect immunofluorescence, depending upon the type of autoantibodies that we are studying. For example, in the case of anti-nuclear antibody, the substrate that we choose will be human epithelial cells. Or in the case of anti-smooth muscle antibody, we take rat stomach. So depending on the type of autoantibody that we are studying, we choose the substrate. So the substrate is usually, the antigen is in the substrate. Okay. Now the primary antibody is from the patient sample. Okay. If the patient is positive, for example, let us take an example of a patient with SLE, systemic lupus erythematosus, which is a very common autoimmune disease. The patient may have antibodies against dsDNA. So in that case, the primary antibody, the patient is positive. So this primary antibody will go and directly bind to the dsDNA on the substrate. Okay. So this is the first step. We give time for, we give incubation time where we allow the antibody to bind to the specific antigen. Then we wash it and all that we do, followed by addition of secondary antibody. The secondary antibody might be usually is an anti-human antibody, which is linked to the dye. So as I was telling you earlier, the dye might be fluorescein isothiocyanate. So then we visualize it under the microscope. So this procedure we'll see in detail in the coming slide. Okay. Now, as such, the basic difference between these two, we know that in direct immunofluorescence, because it is taking a single labeling step, it is a shorter procedure compared to indirect immunofluorescence, which is a two-step procedure. Cost, if you ask me, both are relatively, they are equally expensive. I will not say indirect immunofluorescence relatively inexpensive. No, it is equally expensive. Complexity, if you ask, it is less steps because it is involving one single labeling step, whereas here it is taking an additional step. Sensitivity, if you ask, indirect immunofluorescence is high as far as autoimmune diseases are concerned, whereas direct immunofluorescence, certain aspect, like for example, in this case of Buller's autoimmune disease, this is a gold standard. So it is sensitive in certain aspect, whereas an indirect immunofluorescence is sensitive in many ways. Okay. Now, so we have been talking about immunofluorescence, and one of the most important disease that we are going to discuss is autoimmune disease, which are a group of disorders in which the tissue injury is caused by humoral or cell-mediated immune response to self-antigens. So it is actually a loss of self-tolerance. Actually, if you ask, there are 80 to 100 different types of autoimmune disease and many more. Like, for example, 40 diseases are considered to be having an autoimmune origin. So these autoimmune diseases, during this, when a patient develops autoimmune disease, they develop autoantibodies. These autoantibodies are the important diagnostic markers for autoimmune diseases, especially systemic autoimmune rheumatic diseases, SART. Yeah. Now, so if you ask autoimmune disease, the most important marker will be ANA, anti-nuclear antibody. So from the name itself, we can understand that anti-nuclear antibodies are nothing but antibodies directed against the components present inside the nucleus. But it also encompasses antibodies against cytoplasmic antigens as well. Okay. Now, this, if you see this, this is a picture of a cell where we are seeing this condensed chromosome. If the patient has antibodies, it is going and binding to the this condensed chromosome. And the second step, we are adding the secondary antibody, which is linked to the dye. So this is what is actually we are doing in indirect immunofluorescence technique. Okay. Now, coming to the systemic autoimmune disease, we know that anti-nuclear antibodies are the important marker for many of these diseases, especially SLE, Sharp syndrome, Jogan syndrome, systemic sclerosis, polymyositis, and dermatomyositis. So they are the most important markers for all these systemic autoimmune diseases. Okay. Now, whenever a patient comes with clinical symptoms of autoimmune disease, like the symptoms are quite vague. The clinicians will agree with me. They will have a body pain, they will have knee pain, joint pain, and all that. So whenever we want to screen ANA, there are two methods to screen ANA. One is the ELISA method, that is ANA screen ELISA. And the second method is the indirect immunofluorescence technique. Okay, now what happened was in the past, that is in 2008, they were seeing large number of false negative results in ANA results in USA. Lot of, that is about 35% of patients with SLE were given a negative result. So everybody was little worried because why this is happening. So they created a task force, you call it as ANA task force, to investigate the reason for this observation. So what happened was, so the results of ANA task force was, they understood that they observed that this false negative screening results were mainly achieved with multiplex test using purified antigen. Okay, so whenever a patient is screened, ANA is screened using ELISA method, what happens is, in a microwell, we can code only minimum number of antigens, like 10 to 15 antigens you can code. So if a patient, let us imagine a patient has antibodies against histones, but that histone is not coded in the well, obviously the results will be negative. But is it really negative? No, it's a false negative result. So they concluded that for ANA screening, the ideal method will be indirect immunofluorescent technique, and the substrate that has to be used is HEP2 cells, human epithelial cells. HEP2 cells should be used as a standard substrate for any screening, and definitely the titer should be given. Like for example, if it is a 1+ or 2+ or 3+, or in the case of dilution, some labs report it as the initial dilution that we use for Euroimmune kids is 1:100. It depends on the conjugate, how they standardize the conjugate. So it is 1:100. The initial screening will be with 1:100 when we use Euroimmune kits. So some labs reported in the form of dilution 1:100, 1:320, 1:1000. So giving a titer is very important. And we have to describe the ANA pattern. So the most important aspects that are important, critical in a report is, first of all, we have to write whether it is a positive or negative. Then we have to say what is the method that is used, indirect immunofluorescent technique. And we have to mention the titer or the dilution and describe the pattern. So these are the most important aspects in a report of ANA. So we all know that the clinical symptoms or clinical manifestations play a very, very important role here. It's not only diagnostic aspects, clinical and the diagnostic aspects go hand in hand. So that is very important. So clinicians will decide what type of treatment they have to give depending on the titer of the patients. How much the patient has got titer, depending on that, the clinician will decide. So we have also noticed that some healthy people in the range of 5 to 11%, some papers reported as 18%. So some healthy patients or people also develop ANA. So it is very important that the clinician will look into the clinical aspects. If the patient doesn't have any clinical symptoms, but in case the patient is ANA positive, then the ball is in the clinician's court. He will decide the course of action. So diagnostic aspect and the clinical aspect go hand in hand in the case of autoimmune disease as a whole. So we all know that when there is a wrong ANA testing, so there is a lot of problems. Basically, first for the patient, because it gives unnecessary suffering for the patients, because there is a delay in diagnosis. It might result in wrong treatment, delayed treatment, and definitely for the laboratory, it is going to be a higher cost. Because the patient may show symptoms, we might be giving a negative report, we have to repeat again and again. So it is very important that in the case of ANA or autoimmune disease, correct diagnosis is very, very important or crucial. Okay, so this slide I just wanted to add because I just wanted to show you this indirect immunofluorescence on HEP2 cells. We are able to screen around 100 to 150 potential autoantibodies, antigens. So whenever we give a report with indirect immunofluorescence, it's going to be absolutely correct because it's a very sensitive method. But whereas when we use a screening method like ELISA, we are not very sure because there is a possibility of false negative results because of the limited number of autoantigens. So it is very important. The point that I want to stress is for ANA, indirect immunofluorescence is a gold standard. So we should never do a screening. It's better not to do screening with screen ELISA because there is a risk of false negative result. Okay, now, so we have just talked about autoimmune diseases. We talked about direct immunofluorescence, indirect immunofluorescence. Now we'll see what is this indirect immunofluorescence. What are the steps involved in indirect immunofluorescence? Yeah, so we all have to remember 7S for indirect immunofluorescence. The 7 important ingredients which starts with yes. to remember, to easily remember, I thought I will put it in the S series. So number 1 is a specimen. The specimen that we always use for ANA or ANCA or liver is serum or plasma. So some people, some laboratory doctors will ask whether we can use pleural liquid. No, it is advisable to use simple serum or plasma as specimen for indirect immunofluorescence. But in the case of neurology, yes, definitely you can use CSF. Whenever we use CSF for neuronal study, we use it undiluted. Whereas when we use serum or plasma, usually serum, we dilute it depending on what autoantibody we study. For example, in the case of ANA, as I told you, we use 1:100 dilution. Whereas in the case of ANCA, we use 1:10 dilution. So that changes depending on the autoantibodies that we are studying. So the number 1 S is the specimen, which is the serum or plasma. And number 2 is the substrate. The substrate, as I was telling you, depends on what autoantibodies that we are studying. As I told you, if it is ANA, we use human epithelial cells. If in case we are using neuronal autoantibodies, we use cerebellum. In case we study anti-mitochondrial antibody, the ideal substrate will be rat kidney. Or in the case of anti-smooth muscle antibody, we use rat stomach. So the substrate is chosen depending on the autoantibodies that we are going to study. That is the second yes. Specimen is over, substrate is over. Third one is suitable fixator. So this substrate is fixed onto the slide. So the fixators might be different, like there might be an acetone fixation, there may be ethanol fixation, it might be a formalin fixation. These fixations play a very important role in keeping the substrate intact, and the expression of antigen also matters a lot here. So in the case of ANCA, ethanol and formalin fixation is used. In the case of HEP cells, we use usually acetone fixative. So these fixative play a very, very important role. So that is the third yes. The fourth yes is nothing but the saline water buffer, wash buffer. The buffer that we use here is phosphate buffers, phosphate buffered saline, PBS is a wash buffer that is used. So usually the sample is diluted with the buffer. We don't use water anywhere, but for the preparation of buffer. So preparation of buffer has to be very carefully done, because everything like dilution, washing, everything is done using this PBS buffer. So that is number 4. Saline water buffer, wash buffer. Fourth is the buffer. Fifth one, the fifth S is the secondary antibody. The secondary antibody is usually given in the kit. It is an anti-human antibody, which is usually conjugated to the dye, FITZ dye, ideally for IIFT. So that is the fifth S. And the 7th yes is the slide mounting with the slip. So you have so finally when we are done with the procedure, we put everything on a cover slip. We keep a cover slip, add the glycerol, then mount the slide. We'll be showing you in the coming slides. So the slide fixation or mounting is the 6th important yes here. And finally, once we have finished the processing, it's the see-through. You see it under the fluorescent microscope. So these are the 7 S that is the most important ingredient for indirect immunofluorescence: specimen, substrate, suitable fixator, saline wash buffer, secondary antibody, slide mounting with a slip, finally see-through. Okay, so these are the 7 S that we have to remember. Now, so first steps, I'll just show you this, then we'll see the video. So first step, what we have to do is, you are going to take the sample. So if the patient sample, you can, as I told you, serum is ideally recommended. So if it is an ANA study, we have to do a 1:100 dilution. So first what we have to do is prepare the buffer, PBS buffer. Ideally, the PBS buffer can be used for a week. Ideally, if you ask, it is for a week that you can use. So you have to prepare the buffer. How do we prepare? In the kit, they'll be providing you with the buffer, which is it will be like a salt sachet. What we have to do is take 1 liter of distilled water of very good quality, provided the pH is 7.2. It should not be very less pH, or it should not be high pH, because if there is a difference, the pH is not proper, definitely it is going to affect the performance of the test. Okay, now you are prepared. You take 1 liter of beaker with the distilled water, add this buffer, which is like a salt. You add this 1 gram of salt, which is your PBS, then mix it very well, nicely without any crystals. Then add the Tween 20. Tween 20 is a detergent that will avoid a non-specific staining. You don't get a background. So we need to add this Tween 20. It's a simple detergent. So now our PBS buffer is ready. So PBS buffer, 1 liter of distilled water, add that salt, mix it very well so that there are no crystals, and then you are adding the Tween 20. So then the buffer is ready. Now the first step is we are adding the sample onto the substrate. So the substrate, as I was telling you, is chosen depending on the type of autoantibody. Now the first step, we are adding the sample. So the sample, when it is added, if the sample contains the antibody of interest, it will go and bind to the specific antigen on the substrate. Might be dsDNA or a mitochondria, or a specific like enzymes like estradiol synthetase, anything on the substrate. Okay, it will go and bind. What we are going to do, then we are giving a incubation period of 30 minutes so that the autoantibody will go and perfectly bind to the antigen. It's an antigen antibody reaction. Now we are giving an incubation of 30 minutes. Then we are washing it. Then what we are doing is we are adding the conjugate. Okay. Now we are going to add the conjugate, which is anti-human antibody, which is got tagged with the fluorescent dye. Okay. Now, if the binding is already intact even after washing, this dye will go and bind to it. Okay. Now, after that, we are giving an incubation of 30 minutes again. Then we wash it with the PBS buffer. Then we are mounting it. Then we see it under the microscope. So these are the basic steps that is involved in indirect immunofluorescence. Technically, it takes around 1 hour, 15 minutes, actually. So very simple technique. The point is, but it has to be done very, very systematically, carefully. So whenever we want to start the test, for example, we want to start at 9:30, we keep the kit out at least 15 minutes before so that it comes to the room temperature. We should not just take it from the fridge and start it. It will disturb the complete protocol. So we make sure the kit comes, the ingredients comes to the room temperature. Okay, so that is very important. And we have to make sure we maintain the time correctly. Incubation 30 minutes, we have to follow 30 minutes. It should not be 20, 25 or 35. So the 30 minutes incubation has to be perfectly followed. So I'll just show you the procedure. So I'm just showing you a video of this. Yeah. Yeah. So this is it. First thing, what we do is we take the sample, serum sample, we vortex it very well. Okay, nicely vortex it. Then we dilute it. For example, for ANA, we do a 1:100 dilution. Okay, so either we can take 990 microliter of the buffer and 10 microliter of the sample. So we dilute it and add, this is a titer plane. I will be talking about titer plane in the coming slides. So what we do is we add 30 microliter of the diluted sample. So we add 30 microliter of the diluted sample without any bubble. So for every slide, we do a positive control and a negative control, so that we can come to know that if there is any error, we can see it from the control, control thing. So that is very important. So adding a positive control and negative control is quite important. So after adding 30 microliter, we just invert the slide. So this slide on the lower part has got the substrate. So the substrate is has the antigens, so it will go and bind to the sample. If the sample has autoantibody, the antigen antibody interaction is going to take place. Okay, now so we are just leaving it for half an hour. Okay, now after that we take out the slide, wash it very gently with the buffer. Very gently. We don't use a pasta pipette, we don't use a pipette or anything. The washing is very gentle, and then we keep it in a coupling jar with the buffer. Okay, so what we did, we took out the slide, we washed very gently with the buffer, then we put it in a coupling jar, ideally a glass coupling jar with the buffer, and we kept it for 5 minutes. In the second step, we are taking 25 microliter of the conjugate, we are adding, then we take out the slide. See how he is wiping only the opposite direction, invert it like this for 20-30 minutes, then take it out, then wash it gently again with the buffer, then keep it in the coupling jar for 5 minutes. Then after that, what happens is you take out the slide, only wipe the opposite direction, never touch the front side. Okay, very gently. Then what she has already done is she has taken a cover slip and she has added the glycerol, the mounting medium. It's just like a glue with the appropriate pH, so that this slide can be, see how she is doing, and inverting it on the cover slip with the glycerol, and that is kept intact, and we see it under the microscope. So this is the simple procedure where what is more important is washing here. If we don't wash it properly, we get unwanted background fluorescence, and we have to correctly follow the dilution. 1:100 for ANA, or if it is ANCA 1:10, the dilution has to be followed. Then what is more important is the water that we use for the buffer preparation should be an optimum, should be with optimum pH. So that is all the important things that we have to remember for during this procedure. Otherwise, it's a very simple procedure and easy one. Yeah. So that is how it is done. Okay, so why are we using indirect immunofluorescence? I just want to once again reinforce this point. It is a gold standard for ANA screening. So we are able to study the complete antigen spectrum. We can see cell nuclei, we can see the cytoplasmic antigens. So complete antigen spectrum can be studied. And one substrate, that is HEP2, we'll be able to screen around 150 different autoantibodies. So complete antigen spectrum can be studied and has got a high specificity and sensitivity. So that is why we always recommend to do indirect immunofluorescent technique, because of all these advantages. Okay, now we were talking about the standard substrate for ANA, which is human epithelial cells. What is these HEP2 cells? These are the cells from an epithelial cell line from human laryngeal carcinoma. So they are laryngeal carcinoma cells, which are standardly used as substrate in indirect immunofluorescence. So the purpose of using HEP2 cells, it is number one, it is they're very easy to cultivate, they are easily standardized, you have a good number of human antigens, and you have a very large cell nuclei and good proportion of mitochondria. So these are the reasons for which HEP2 cells are chosen. Easy to cultivate, can be easily standardized, good amount of nuclear cell nuclei, and good proportion of mitochondria. So that is why HEP2 cells are standardly used as substrates for any indirect immunofluorescence. Now, so we've been talking about Hep2 cells, but Euroimmune has Hep2010 cells. So there is a difference. Like when you see this left-hand side, you are seeing only 3 red circles, whereas when we see right-hand side, we are seeing many red circles. It is actually nothing but in the right side picture, you're seeing more number of dividing cells. So the more dividing cells or more number of mitotic cells will help us in identifying patterns that includes mitotic cells, like homogeneous pattern, or if it is mitotic patterns. So the patterns that includes mitotic apparatus can be very well distinguished with this Hep2010 cells because of the more number of dividing cells. So Hep2010 has got that advantage, 10% more mitotic cells compared to Hep2 cells. Okay, now these two technology, I just wanted to brush it up, biochip and the titerplane technology, which are the patented technology from Euroimmune Germany. I'll just brief you on that. We have been using a conventional method. What do we do? So this is a slide which has got many substrates like stomachs, substrate, liver, kidney, intestine, lungs. So when we want to study a patient's autoantibodies, what do we have to do? We have to add sample here, here, here separately, which will take a lot of time and effort. So every time you have to add the sample, you have to study it separately. Just imagine having all these substrates in one field. Like in one field, if you have stomach, liver, kidney, intestine, lungs together, then it's a very big advantage. You need to add the sample only once you're adding, and you can study the spectrum of autoantibodies in all these substrates, which gives us a comprehensive profiling, which is multiplexing. So that is the advantage of using biochip technology. So how do they do it? They take a tissue, they slice it very nicely, and they fix it on a glass slide. Then they cut it into biochips, and the cover slip, which has got the full complement of the tissue, is taken. Then they put it on the, see, this is how they do it. They put a glue, and they just add these biochips. It looks very simple, but it's a very tedious process. See, this is a slide, and they take these biochips and they paste it. So in one field, so this is the slide. Usually Euroimmune slide will have a Euroimmune name here, and it will also have a name of the substrate that is being used. So this is called a field 1. There are 10 fields here. In one field, we are seeing some 6 windows, right? 6 substrates are there. So 6 substrates are there. So it can be any type of substrates. It can be a tissue, or it can be a cultured cells. It can be a transfected cells. It can be a purified antigens. So any combination of substrates is possible and feasible depending upon what we are studying, what autoantibodies we are studying. So like in the case of liver, we have a comprehensive 6 substrate like Hep2 cells, primate liver, rat kidney, rat liver, rat stomach, and VSM. So where we are able to comprehensively study the complete autoantibody spectrum in the patient. You're adding sample only once. Okay. So, so here you have 10 fields. That means you can add positive control, say like a negative control, and you can add 7 samples here. Oh, sorry, 8 samples here. Okay, so if in case a customer, if you have a very lower load, that you have only 1 sample, you can add a positive control, negative control, 1 sample. Or if you have 3 samples, you can choose a 5 field slide. So depending on the requirement, the slides are available. 3, 5, or 10 fields are available. Okay. Now, so that is the advantage. So combination of cultured cells, tissue sections, purified antigens in the same test field. So all these, and you can also combine substrates with different fixative, and it's ideal for high throughput and multiplexing. And coming to the tighter plane technology, see, it's a very important technology as far as IFT is concerned. See, this is what I wanted to show you. This is the tighter plane as we have seen. So the point one is here in this case, you have a hydrophilic interior and a hydrophobic exterior. So when you add this sample, the sample, it just holds on like that as a bubble, doesn't come out. So there is no cross contamination, and there is uniform incubation period. Uniform incubation period is maintained. And if you are not using this tighter plane, if you're using a slide as such, there is a risk of the substrate being, I mean, what you can say, it's disturbed when you use a pipette. All that problem is solved when you use a tighter plane, which is reusable. You can use it 3 months depending on the workload, and you can just wash it with distilled water. So it comes free of cost. So this is the tighter plane technology. So whenever we use indirect immunofluorescent technique, the tighter plane is a must. Okay, so that is what I wanted to tell you. So it's easy handling, simultaneous start of all the reactions, drops don't spill over, no cross contamination. You don't require any humidity chamber. When we don't use a tighter plane, there is a risk of sample drying out. So what happens? We have to use a humidity chamber to avoid drying of sample. So all that can be avoided. So this is big. This is a very important technology from Euroimmune. So biochip and tighter plane. Yeah. Now coming to ANA strategy. So as we all know, there are two. It is a two-step strategy. First is screening. We have to screen it with a multi-specific test like indirect immunofluorescence. If the patient is positive, it has to be confirmed by another method like a blot or monospecific ELISA. Then we release the result. Okay. Now, in the case of patients who is negative, we don't have to do a blot or a monospecific ELISA. So that is the two-step strategy: screening and then confirmation differentiation with monospecific assay like blot or ELISA. We should not directly go to a blot, because in blot you have a limited number of antigens. You have around 15 or 18 antigens or 23 antigens, only 23 maximum. But if the patient has got antigens apart from these 23, we are going to get a negative results. We are not sure whether it is actually negative or it is negative for these antigens. So it is ideal that we screen the patient with indirect immunofluorescent technique first. Then if it is positive, we go for a confirmation or differentiation with blot or monospecific ELISA and release the result. Okay. Now, what is a substrate? So we were talking about HEP2 or HEP 2010. So what the biggest advantage with the Euroimmune is you have two substrate combination. One is HEP 2010 and primate liver. So two substrate in one field. So in one field, you have two substrates. See, the advantage is we can confirm certain patterns. So we are not very sure what pattern is. So this primate liver can give you an indication of confirming, or maybe this is this pattern. And some second important point is differentiation. Like certain patterns look similar in HEP cells, but they look different in primate liver. So that is the advantage of having a primate liver along with HEP cells. So whenever we start visualizing the slide, first we should see HEP cells, then we decide what pattern it is, then we go to the primate liver for better understanding. Like we can differentiate, we can confirm. So all these can be done with the help of primate. It's an additional substrate that gives you little more information. Okay, so that is the advantage of having two substrate combination. Yeah. So this slide I just wanted to tell you, because anti-nuclear antibodies and antibodies can be against any of the components in the cell. It can be against mitochondria, it can be against lysosome, it can be against Golgi, anything. But the function of this particular component is compromised. Okay, so this is a broad classification of ANA, where depending upon where you see the fluorescence. For example, if you see fluorescence in the nucleus, you call it as a nucleus pattern. If you see the fluorescence in the cytoplasm, you call it as cytoplasmic pattern. If you see the fluorescence mitosis, you call it as mitosis pattern. So actually, these are the broad classification for ANA. So for nucleus, I'll just briefly talk about this. So for nucleus, we have 5 classification. One is homogeneous. Second one is speckled or granular. Number 3 is dotted. Number 4 is nucleolar. And then we have the membrane. So in the nucleus pattern, we see only the nucleus. We don't see the cytoplasm at all. So how you see the fluorescence? Like, is it speckled? You call it as speckled. If it is completely same or homogeneous, you call it as homogeneous. If you see beautiful dots on it, you call it as dotted. If you see the nucleolar accentuation, we call it as nucleolar. If it is only the membrane taking the stain, we call it as membrane. So 5 basic classification under cell nucleus. Now coming to cytoplasm, there are only 3 classification. That is granular, fine granular, filamentous. Here we see only the cytoplasm. The nucleus may be dark or it might be faintly visible. So the cytoplasm, how it looks, whether it is coarse granular or it is fine granular or it is filamentous in nature. So that is how you call it as cytoplasmic fluorescence. The next category is mitosis, where you see the mitotic apparatus coming up, like a spindle or the centriole or the mid-body or condensed chromosome. So these are the basic categorization as far as ANA patterns are concerned. I'll just show you the most important antigens here, because we cannot take everything in this short period. So I've just showed you a picture where a slide which has all the information on the antigens. Okay, when it is a homogeneous pattern, the most important disease is SLE, systemic lupus erythematosus. So when a patient develops homogeneous pattern, the antigens can be dsDNA, nucleosomes, histones, or ssDNA. So 4 antigen can be there if the patient develops homogeneous pattern, and the disease is SLE. Most important antigen will be dsDNA. Okay. Now, if the second category, or you call it as granular or speckled one, where we have DFS70, NRNPSM, SSA, SSB, COO, Me2, PCNA, mitosis. There are so many there. I've just highlighted the most important ones. See, the antigens can be named after the biological function. Like, for example, ribonucleoprotein, or it can be named after the patient or Smith, or it can be named after the disease like SS, Sjogren's syndrome. So it can be named after this function. It can be any way it can be named. So when it is dotted, it is few dots, few nuclear dots, centromeres, depending on the number of dots you see. Okay. Now it is nucleolar. We have 5 types, 5 antigens: SEL70, PMSEL, fibrillary, RNA polymerase, NOR90. So these are the nucleolar antigens. So these nucleolar antigens are mainly associated with a disease called progressive systemic sclerosis or scleroderma. So all of them mostly are associated with PSS. Okay, now coming to membrane. So only the membrane are stained. So basically these are antibodies against the antigens present on the membrane. They are basically receptors like lamins, or it can be a nuclear pore protein like glycoprotein 210, which is the molecular weight of glycoprotein, or P62. So these are the antigens associated with homogeneous pattern, speckled pattern, dotted. Dotted is actually, we call it we are giving a pattern name because many antigens are involved. So nucleoli and membrane. This membrane is antigens are mainly associated with the disease PBC, primary biliary cholangitis. Okay. Whereas in the case of cytoplasm granular, we have anti-mitochondrial antibody. So immediately we should think about the antigens present in the cytoplasm. That is organelles like mitochondria, Golgi, or the lysosomes. Jo1 is nothing but the synthetase, tRNA histidyl synthetase, named after the patient John. Okay, so these are the most important granular antigens, whereas the fine granular includes ribosomal P protein, whereas the filamentous is cytoskeletal proteins like actin, vimentin, tropomyosin. We also have vinculin. We also have rings and rods pattern. So these are under cytoplasmic antigens. Whereas in the case of mitosis, they are spindle fibers, centrioles, mid-body, and condensed chromosome. So this is a broad classification of anti-nuclear antibody, depending on where we see the fluorescence. So whenever we see it under the microscope, first of all, we have to decide what we are seeing. We have to, first of all, see whether we are seeing the nucleus pattern. If not, if it is a cytoplasmic pattern, okay. If you're seeing a nucleus pattern, how does it look? Is it homogeneous or is it speckled? Is it dotted? Are you seeing the nucleoli or the membrane? So we classify accordingly. In case we are not seeing the nucleus, we are seeing the cytoplasm. How the cytoplasm is taking the fluorescence? Is it granular? Is it fine granular or filamentous? So we have to segregate into different types depending on how we see it under the microscope. So we should never write the name of the antigen when we are doing this indirect immunofluorescence, because it's a screening technique. We have to just know what pattern it is. We have to just specify whether it is a granular, whether it is homogeneous. We should not write it as dsDNA unless and until we confirm it. So that is important. But when we know what antigen it is a very big advantage, because in case we know that what we are seeing is Q, then it will help us to decide on what is the blot we are going to take. We'll choose the blot which has that particular antigen. So for that aspect, when we know what antigen it is by indirect immunofluorescence, then it becomes easy for us to choose the right blot. Otherwise, what will happen? Your IFA will be positive, blot will be negative, will be confused. So that is a problem. So that gives an advantage if we know what antigen is that in IFT. Okay. Okay, I'll just touch on the most important patterns. So this is homogeneous pattern. What we are seeing is only the nucleus. The cytoplasm is completely dark here. The nucleus is homogeneously stained, and we are seeing this condensed chromosomes. So these are the condensed chromosomes. So two aspects are important. This is Hep cells, this is primate liver. So here two aspects are important. One is the nucleus, homogeneously stained nucleus, and the condensed chromosome. If we don't see the condensed chromosome, then this may not be classified under homogeneous pattern. So these two aspects are important. And number three, the primate is always positive here. Because the primate liver cells are very small, the nucleus is really small here. Okay, so these three aspects. One is homogeneously stained nucleus, condensed chromosome, and primate liver taking the stain. Three aspects are important to know that this is a homogeneous pattern. So what do we do next? So as I was telling you, there are a lot of antigens like dsDNA, nucleosome, histones, ssDNA. ssDNA is a non-specific antigen. It comes in many diseases. So the most important target antigen will be dsDNA. So the dsDNA can be confirmed by three methods. One, we have an IF method, which is the criteria method, which is actually a hemoflagellate, which has got a nucleus. This is kinetoplast. You have a basal body and a flagellum. So what is more important is it has to be positive in the kinetoplast. If it is not taking fluorescence in the kinetoplast, it is negative. So this is a criteria method, which is highly specific, but it may not be that, what do you say? It's not that sensitive. The best method will be ELISA method, which helps in quantification, and the dsDNA NCX will be highly sensitive compared to the blot, or it is RA and all that. So dsDNA, the best method to confirm will be NCX method. Okay, so this is about dsDNA. So I've taken one image of speckled pattern. So one of the important antigen is NRNP-SM, ribonucleoprotein. So you can see this nucleus, speckled nucleus. It's not homogeneous. It is granular, coarse granularity you can see here. And other feature you can see, this chromosome is unstained. The nucleoli is also unstained. So the cell is in the telophase. Telophase stage is about to divide. Okay, this is in the metaphase stage. You can see this chromosome is unstained. So these three aspects are important. One is coarse granularity of the nucleus, and the chromosome is unstained, nucleoli is unstained, and primate is always positive in the case of NRNP-SM. So there are many antigens for which a primate might be negative. So it is not always mandatory that primate has to be positive. But what is more important is Hep cells. What we see in Hep cells matters a lot. Primate gives an additional information, that's all. So this is NRNP-SM, which is a very important antigen in the case of mixed connective tissue disorder, MCTD. Okay, this I wanted to just show you. This is PCNA. PCNA, where we have only 20% of cells taking the stain. The cells which are bright, here and there you can see some bright cells, and those cells you cannot see any dividing cells. These are all dark actually. So these are only few cells taking the stain. Primate actually doesn't happen. It is negative actually. For PCNA is absolutely primate is negative, whereas you can see 20 or 25% of cells taking the stain. It is a very important pattern or specific for SLE, though the prevalence is quite less. So PCNA is proliferating cell nuclear antigen, or you call it as cyclin 1. Okay, so this I just wanted to show you. This is a granular chromosomes, right? You can see the nucleus, which is granular, and the chromosome is also, see, this is, and the nucleus is granular, chromosome is also granular. So this I wanted to show you because we are talking about a very important antigen, which is DFS70. Dense, fine, speckled 70 is the molecular weight. So this is how it looks. See, the nucleus is completely speckled, and we can see this condensed chromosome. The condensed chromosome is also speckled. So whenever we see condensed chromosome, we always think it is homogeneous pattern. But what happens is, this is a different antigen called DFS70, where the nucleus is speckled. So we see two things. One is speckling of nucleus, and number two, the chromosome is also speckled. So two aspects, and most of the time the primate is negative or it might be weak. Whereas homogeneous is, primate is always positive. So that way you can differentiate homogeneous from DFS70. But the most important aspect here is the nucleus is granular, as we see here. This DFS70 is important because it can be an exclusion marker for rheumatic disease. It is usually seen in some healthy people, atopic dermatitis and all that. When this is seen, the patient, you can exclude it from patients, to have SLE or Sjogren's. So it is very important that we should not make a mistake it with the homogeneous pattern, which is very it's a marker for SLE, whereas this is an exclusion marker. So we need to be very careful because we see speckled nucleus and the chromosome is also speckled here. So that is DFS70. So coming to the next thing, which is the dotted, the number of dots inside the nucleus is important. Like for example, we see 5 to 20 dots. It is called nuclear dots. If you see 2 to 6 dots, it is few nuclear dots. If we see more than that, 46 to 92 dots. See, this is a nucleus which has got 46 to 92 dots. Here, this chromosome, this is the centromeres. So it's a very beautiful pattern on the Hep cells. Most of the time primate is, you can see very minute dots here. Otherwise it's negligible. So this is one of the very beautiful pattern. Maybe you don't have to do a confirmation. If you're very sure about the pattern here, you can directly report as centromere. This is centromere, which is 46 to 92 dots. So coming to nucleolar. So nucleolar, what we see the nucleoli, beautiful nucleoli. You can see nucleolar accentuation, This nucleus, what we are seeing is the nucleus. Inside that, we are seeing this nucleoli. So this is nucleoli pattern. Okay. Whereas primate also, you can see the nucleoli, which looks like a small dot. So this is actually SEL70, or you can call as anti-topoisomerase 1, which is a marker for PSS or scleroderma. I just wanted to show you how different patterns look. So they've taken the most important ones. So coming to the cytoplasm, in the cytoplasm pattern, the most important one will be anti-mitochondrial antibody. So here, the nucleus is completely dark, whereas you can see this cytoplasm nicely taking the coarse granularity. You can see the cytoplasm nicely taking the stain. Nucleus is completely dark, whereas the primate is granular. So this is a very, very important antigen as far as PBC is concerned. 95% of patients with PBC will have this particular pattern. So it's a very important pattern for autoimmune liver disease like PBC. The best substrate for AMA will be rat kidney. Ideally, the gold standard substrate will be rat kidney. So in Hep cells, you see this beautiful extension of the cytoplasm, very nicely seen. This is cytoplasmic fine granular, where the cytoplasm is homogeneous. It looks like a very fine granule. You see nucleoli inside sometimes, whereas a primate looks positive, but most of the time at low titer doesn't take it up. But you can see this fine granules in the cytoplasm. And this is very specific for SLE. Patients may develop neuropsychiatric symptoms if they have this antigen, ribosomal P protein. And then we have this filamentous, as I told you earlier. This filaments, you'll have beautiful filaments in the cytoplasm. Like this is one of the most classical antigen in cytoplasmic pattern. This is lot of needles we can see, like bristles we can see. And primate gives you a beautiful pattern. It gives you like, looks like a spirochete, and this looks like an aeroplane for me. So some beautiful pattern you can see. This is actin at Hep cells. In Hep cells, this is primate liver. This is a very, very important antigen for autoimmune hepatitis type 1. So the point about actin is we have to confirm actin by another IFT method only. So because it is a filamentous actin, we cannot coat it in a blot or ELISA. So as per international consensus, confirmation of actin should be done by another IFT method. Might be the substrate is VSM47, vascular smooth muscle, which is a substrate used to confirm actin. So this is actin, beautiful. It looks so beautiful on Hep cells and primate liver. So mitosis, what we see is spindle apparatus. You can see the beautiful spindle, centrioles, mid-body condensed chromosomes. So these are the most important categories and the antigens as far as the nucleus, cytoplasm, and mitosis patterns are concerned. So I just wanted to tell you about this ICAP, International Consensus on A&E Patterns. So what happens is like different labs report sometimes differently. For example, if a patient has a cytoplasmic pattern, some of the lab may report it as ANA negative, cytoplasmic positive. Some lab report even cytoplasmic patterns as ANA positive. So same patients, different report. So in order to avoid that, in order to bring standardization and reporting, so this ICAP has come out with numbering for each and every pattern. So this is a broad classification of the ANA, right from AC0. If it is a negative also, it is given a number. AC is anti-cell. So antibodies against the cellular components. So every pattern is given a number, right from AC1 to AC29. So the nuclear pattern is given a number from AC1 to AC29. So AC14 and AC29. So we have to remember one thing here. The topoisomerase, which was considered in nucleolar pattern, is under speckled pattern if we are following ICAP. So this ICAP, this cytoplasmic is from 15 to 23, whereas mitotic is from 24 to 28. So there are two challenges when we use ICAP. One is the clinician should be aware about this ICAP numbering. Number two, we should be thorough about the pattern. See, for example, unless and until we know that this is anti-mitochondrial antibody, we cannot give AC21. But when we are following the earlier classification, whether it is Heme or Golgi, we give it a cytoplasmic granular. Whereas when we are following ICAP, you have to be very specific. If it is Golgi, we have to write AC22. Like that. So homogeneous is AC1, DFS is AC2, centromere is AC3. So this is very important when we are talking about ICAP consensus. But in India, we are not still we are not following this ICAP right now. Maybe it will take some years. This will give will help in standardization and reporting actually. Dr. Kalita, do I have time for ANCA? Do I have time for ANCA? I have some 5 slides on that. Can I continue with ANCA?
Yes, please continue.
Yes. So, so this is see this ANA to study in detail. If you have any queries, you can write to us. We have a regular webinars happening every month. We have 2-3 webinars every month. So every pattern we'll discuss in detail. We also have a residential program. Because of the COVID pandemic, we are not having it. Okay, so this is a brief thing about ANA. There are a lot of antigens coming up daily, I can say. So coming to the next important autoantibodies, that is ANCA, anti-neutrophil cytoplasmic antibody. So, as far as definition is concerned, they are a group of antibodies against antigen in the cytoplasm of neutrophil granulocytes. So when we take this neutrophil, this is a multilobe nucleus, a lot of granules are present in the cytoplasm. So antibodies are directed against these antigens in the cytoplasm. So these are very important. So they are called as anti-neutrophil cytoplasmic antibody. Okay. Now, there are 2 types of ANCAs. So for ANCA, what we do is the substrate that is chosen is granulocyte, because we are studying antibodies against granules or antigens in neutrophil. So we take the substrate as neutrophil or the granulocytes and we fix it by ethanol. So I hope you are remembering the third S, which is a suitable fixator. Here the fixator is ethanol. So depending upon where we see the fluorescence, it is classified as C ANCA and P ANCA. So C is nothing but cytoplasmic ANCA, where we see the fluorescence in the cytoplasm of the neutrophil. So beautifully we can see the cytoplasm taking the stain. Whereas P ANCA, perinuclear ANCA, we have the perinuclear fluorescence, just like a popcorn. Okay, it looks like a beautiful popcorn. So C and P, only 2 types. Whereas in ANA, we have a lot of antigens. Here, only 2 most important 2 patterns we have, C ANCA and P ANCA. Now, so the point that we have to remember here is the dilution. The initial dilution is 1:10. Whereas for ANA, Hope we all remember 1:100. Okay. When we go for a lower dilution in ANA, for example, if you go for 1:40, there is a risk of false positive results. Okay, so false positive result when we go very low dilution, 1:40. Whereas in the case of ANCA, we should start with 1:10 dilution. Okay, now if we take the slide, there are 3 substrates as far as ANCA is concerned. One is the granulocyte fixed in ethanol. First field will be granulocyte fixed in ethanol. The second, down to that, is granulocyte fixed in formalin. And adjacent to that, we have Hep2 plus granulocyte fixed in ethanol. So 3 substrates are there for ANCA. So I'll tell you what is the importance of using these 3 substrates. So one is it gives us a lot of information and gives us more confidence in reporting and minimal technical effort. So I'll just go in details. See, I will just see what is the C-ANCA. How does C-ANCA look under the microscope? C-ANCA, cytoplasmic ANCA. So the most important target antigen for C-ANCA is proteinase 3, PR3. Okay, PR3. And the disease associated is GPA, earlier called as vaginous granulomatosis. So GPA is the most important disease associated with proteinase 3. So as I was telling you earlier, there are 2 types of fixation. One is ethanol fixation, the other one is formalin fixation. In the case of ethanol fixation, see, it's a flexible fixation. The antigen can move freely. Whereas formalin fixation, it is intact. The antigens cannot move. They are held to that particular position. We all know that. So this is a 2 different type of fixation. So the pattern looks a little different in there. Like for example, if you take this PR3, first of all, whenever we start with ANCA, we should start with the granulocyte fixed in ethanol. That is a standard substrate. So when we see this, what we are seeing is a cytoplasm taking the state. Yes, beautiful cytoplasm is taking, and that too it is granular. So 2 aspects, granular fluorescence and cytoplasmic fluorescence. Okay, now we go to the formalin. In formalin, we usually don't see the pattern. What we see is whether the fluorescence is positive or negative, whether it is taking the fluorescence or not. So for always PR3, the formalin fixed granulocyte is positive. So these 2 will be positive. So it is in ethanol, we see granular cytoplasmic fluorescence, whereas in formalin, it is always positive. So these 2 will always be positive. But the third substrate, we might be wondering, why do we have this third substrate? Because it has got Hep2, when we have a cross-contamination or cross-reactivity with ANA, we will come to know. Because in case if we get a anti-mitochondrial antibody in the patient, again here the cytoplasm will be positive, but our formalin will be negative for all the ANAs. So that is a very important point that we have to remember. All ANAs will be negative in formalin except lysosome. Lysosome will give you a faint, it will give you a weak fluorescence in the formalin. Whereas all the other anti-nuclear antibodies will be negative in formalin, and Hep2 there it will catch up your ANA. So that is a point why we have this Hep2 here. We will be very careful to we'll be very careful when we see a ANA in Hep2. So we will not be in a hurry to report ANCA. So one more thing about ANCA, what we have to remember is, when we do the processing for ANCA, we have to report it as early as possible, within 2 hours or within the same day, because ANCA, the antigens are not very stable. It will just disperse. For example, P-ANCA. So ideally for ANCA, we have to report it on the same day, ideally 2 hours. So this is how PR3 looks. Cytoplasmic granular fluorescence. Here this is positive. Here we can see the ANCAs here. They are very small compared to the substrate. So next is P-ANCA. P-ANCA is of 2 types. One is formalin resistant, the other one is formalin sensitive. Formalin resistant, the most important target antigen will be myeloperoxidase or MPO. It's a very important antigen as far as the disease MPA is concerned. And you have the other type of p-ANCAs, which is formalin sensitive, or you can say formalin will be negative, or they call it as the atypical ANCA. Nowadays, they are giving the name DNA ANCA, like antigens like elastase, cathepsin G, lactoferrin, BPA. BPA comes here also. So this is p-ANCA. p-ANCA 2 types. MPO is most important one. The other one is non-MPO, which is elastase, cathepsin G. These non-MPOs, which is these elastanes, these are all quite important for disease like ulcerative colitis or the disease like primary sclerosing cholangitis. So this MPO and PR3 are important markers for ANCA-associated vasculitis. Okay, so it's MPO and PR3. Other than MPO, PR3, these antigens like elastase, cathepsin G, they are important as far as CIBD is concerned, chronic inflammatory bowel disease. Okay, so this is about MPO. MPO, I'll just show you how it looks. So this is how myeloperoxidase looks in ethanol. So it gives you a perinuclear fluorescence. And here, as I was telling you, formalin will be positive. So we should not look for pattern here because it also looks almost the same for your PR3, but this will be positive. So again, just like PR3, these two substrates will be positive. The only difference is this will give you a perinuclear fluorescence, whereas in the case of PR3, it gives us a cytoplasmic fluorescence. So with ethanol, we take a decision, or we decide whether it is a cytoplasmic or perinuclear. Then we go to formalin. Formalin, we expect positivity for PR3 and MPO. Then we look at HEP2 and granulocytes to see whether ANCAs are there are any ANA interference, so that we don't make mistakes. So this is how our MPO looks on the slide. Yeah, so this I wanted to show you. Yeah, oh, sorry. So the there might be a situation where we have ANA and p-ANCA, or it's only ANA. So if we have a combination of ANA and p-ANCA, see this ethanol can give you a picture like this. It will take inside the nucleus, and because you have p-ANCA, it will give you a perinuclear. But formalin will be positive because you have a MPO here. Whereas if it is only ANA, the formalin will be negative, whereas HEP2 will give you beautiful fluorescence here. So that is how we can differentiate if it is only ANA or if it is a combination of ANA and p-ANCA. So this HEP2 plays a very important role in this aspect. So this DNA ANCA, or called as atypical ANCA, they have an important role to play in ulcerative colitis. So I just want to hint on the consensus, ANCA consensus. The earlier consensus, that is 2003 consensus, they were giving importance to IFT, screening with IFT first, then confirmation with monospecific ELISA or plot. But the recent consensus, 2017 and 20 consensus says that we have to do a monospecific ELISA or ELISA in the beginning. But what is the problem with this is they have considered only ANCA-associated vasculitis. So when we do only monospecific ELISA, we will miss, in case if the patient is positive for this DNA ANCA, we will not be able to know. So we will only report it as negative for PR3 and MPO. Maybe the patient is negative for vasculitis, but maybe the patient is positive for ulcerative colitis or PSC. We might miss. So ideally, doing IFT and this monospecific ELISAs in parallel will give you a higher hit rate. So this is a DNA ANCA. It gives you a perinuclear fluorescence in the ethanol, whereas formalin will be negative. It is only the cells that takes up the stain very less, whereas here it gives you a perinuclear. So the point about MPO and DNA ANCA is only this formalin. That different, there will be light difference here. Otherwise, formalin will be negative. So the take-home message is PR3 and MPO, both of the antigens will be positive in formalin, I mean granulocyte fixed formalin. So this CNCA, the most important target antigen is PR3. The most important target antigen for PNCA is MPO, which is formalin resistant. That means fluorescence will come in the formalin slide. Okay. So that is the end of the session. I have a few questions for to make it more interesting. If you know the answers, you can type the answers in the chat box if it is not a problem. Can we go for that? Or you can just answer me. You can unmute and answer me. Don't worry if it is a mistake. Only 5 questions I have. Yes, the first question is, the additional substrate primate liver helps in or aids in removing false negative, gives better results, differentiation confirmation of pattern, or helps in pattern identification. See, what is the answer? Just type the answer. So what is the advantage of having primate liver? Anybody is typing?
Actually, what is happening is
Yes.
we are not receiving inputs from the participants because the systems they have done. I mean, but still they will answer these questions and they will send in their answer. I mean, this chat box, which
No
we
problem.
can always look into it. But please, can you please, Doctor, carry on
Yes.
your queries.
Okay.
Yeah.
So the answer is differentiation confirmation of patterns. So primate liver helps in additional, the substrate helps in differentiation confirmation of patterns. So the second question. Yeah. So the prominent antigen for homogeneous pattern is, so it's a very easy answer. The answer is dsDNA. Yes. Sorry, that's antigen for dsDNA. Homogeneous pattern is dsDNA. Now the third question is DFS70. Under which category does it come? Is it a homogeneous pattern, or is it dotted? Is it nucleoli, or is it speckled? So DFS, dense fine speckled. So it is speckled pattern, or you can call it as nucleus granular. Yes. Now, what is the disadvantage of doing ANA by ANA screen ELISA? Is it false positive results, or is it giving you correct results? Is it false negative results, or is it true positive results? It's false negative results. The answer is it gives a lot of false negative results. Yes. So the most important target antigen for PNCA is, so the PNCA is, target antigen PNCA is, PNCA is MPO. Yes. So that's all for now. Thank you very much. Stay healthy and take care. Just see the questions. Yes. If there are any questions, you can just ask us.
Yeah, actually the questions will come after. We have noted down the questions basically.
Okay, sir. You can just.
Dr. John will now start his
Yes, sir.
his take on this presentation, which is the expert comment or anything else if you want to put. So after that, we'll take up the questions. We have some questions actually. We have received
Yes,
the question. I will just relay those questions.
Yes, sir.
Dr. John will take over now
Yes, sir.
for his
Thank
comments
you.
and for his inputs.
Thank you. Thank you. It was a very concise and informative overview. And definitely, especially the PNCA, I also learned a few points. Thank you for that. And definitely, I think it was very useful for our students. I'll just summarize. I will, I can share the screen, no? How do I share the screen?
Yes, sir, you can share. No problem. You can
There
check.
is this option called share screen, sir, in the down. You can just click that.
Yeah, I looked at it.
Yes. Yes.
Ah, share screen. Okay.
Yes,
Yes.
down below.
Yes. Can you see the screen?
Yes, it is visible.
Yeah. So just to summarize what has been told now, I will just say that ANA by IFA has to be ordered only when systemic autoimmune rheumatic diseases are clinically suspected. They should not be routinely tested in ANA by in normal individuals, those who come with vague aches and pains or something like chronic fatigue syndrome, backache, osteoarthritis, and rheumatoid arthritis, which has been suspected clinically. Also, I would just like to highlight where ANA is useful. ANA is definitely helpful in the diagnosis of SLE, systemic sclerosis, polymyositis dermatomyositis, Sjogren syndrome, juvenile rheumatoid arthritis. It is required. It is one of the guidelines for diagnosis of, it is required a test for the diagnosis of drug-induced lupus, autoimmune hepatitis, and mixed connective tissue disorder. It is not useful for rheumatoid arthritis, multiple sclerosis, that is the neurological disease, multiple sclerosis. It should not be suspected with systemic sclerosis. Confused with systemic sclerosis, which is where ANA is useful. In multiple sclerosis, ANA is not useful. In thyroid disease and idiopathic thrombocytopenic purpura. And just so that the students know, mitosis, ANA detects interphase and metaphase. These two contribute to the patterns. A few patterns such as spindle are based on the anaphase. So we should remember that interphase is the resting cell, and ANA by HEP2, the other point which I would like to reiterate amongst the other reasons for using HEP2 as a substrate is all these cancer cells, they multiply rapidly. So at any given point in time, you have various cell cycle stages of the mitosis, and at different cell stages, different antigens are produced. So IFA with HEP2 cells, which are rapidly multiplying cells, which are a large nucleus, and because they are rapidly multiplying, there are various cell stages, and at various cell stages, various antigens are produced. The chances of detecting the autoantibodies are the highest. So this forms the basis and the cornerstone for using immunofluorescence for detection of ANA. Whereas in the ELISA, you have to quote the different antigens. If you miss some antigens, it will give a negative. So you get a false negative. Moving on, this is just to show. You dilute the serum, IgG antibodies bind, then you add the fluorescent anti-IgG antibody, you wash and you observe under the microscope. The 5 major patterns are the homogeneous, the speckled, the peripheral, that is a antinuclear membrane, nucleolar, and the centromere. So finally, the points I would like you to remember from today's workshop is: 1, the gold standard for ANA detection, the gold standard test for detecting anti-nuclear antibodies is IFA. In addition, it also detects antibodies to other cellular components, that is the cytoplasm. ANA can be detected in the normal population. So one should not test individuals without indication. And definitely it is not indicated in those with fatigue or fatigue-like symptoms, back pain, osteoarthritis, and rheumatoid arthritis. High titers are definitely more meaningful, but as you know, In a diagnostic lab, they are not very practical. So a 1:100 screening is used, followed by specific testing. Another point is, do not repeat a positive ANA-IFA test. ANA is not good for monitoring disease activity, as levels of the antibodies will fluctuate irrespective of the disease activity. Repeat an IFA test for ANA only when autoimmune disease is still being suspected and the initial test was negative. To conclude, ANA-IFA is very sensitive. The biochip format makes it more specific. That is, it reduces subjectivity, but it does not eliminate it. It reduces the subjectivity of the reporting if only we had used HEP2. But HEP2 and the primate liver makes it more specific. For confirming antibody specificity, enzyme immunoassay or line immunoassay or a chemiluminescence immunoassay test is negative. Finally, ANA can be negative and specific test positive, but these are very, very uncommon. Thank you.
Thank you so much, sir.
Dr. Kalita, yeah.
Yes, sir. Thank you so much for your very useful feedback. What I can see actually is many of the questions are answered by your presentation already. I'm talking Dr. Johnson. So there were some questions which I think those questions do not need any answer now. You have answered it already by your very small presentation. But I want to thank Dr. Sunita as well, because it was a great presentation and very difficult and complicated topics. He has made it simple. But having said, yes, sir.
No, yes, I fully agree with you.
Thank you very much, sir.
And thing is, what I can see from the questions itself, that the kind of participants we had here or we have right now, it's a very different categories of people are there, and some of them are just a beginner, or they want to start the thing in their respective centers. Some are already into it for some days, and some people are advanced user, very few of them. So from the question pattern, I can make out that. And I hope that the presentation was very, very useful for everybody, especially for the beginner, because there are many things. Actually, these workshops should work as a launchpad from them, and they need to. Just like Dr. Sunita told, that they run some workshop commonly, so they can take help of that, and they can further proceed for their ultimate goal of reaching some good quality level. Like what I have seen in Dr. John's lab, it was very well equipped lab for IFA and autoimmune disease lab. Was very great. Actually, I had the opportunity to see it very briefly, but it was very inspiring for me. And now I actually go to the questions. There are some questions we have received, and one of them was from, what is her name, Dr. Nishanti. And she was asking of the secondary antibody. This question is for Dr. Sunita.
Yes,
Secondary antibodies. So she's asking which animal basically origin antibody is this? The secondary antibody, that's ANA they are talking about.
Goat. They use anti-human antibodies, mainly this mouse, goats, sir. Goat antibodies.
Yeah.
Okay.
Just to clarify, what happens, what is an antibody in the human, when you take and give it into another animal, it acts as an antigen there. So you will get an antibody against the antibody. So that principle is used in immunology to get the secondary antibody, which can be tagged with a reporter, either an enzyme or a dye or a chemical agent which gives luminescence. So with this, this principle is used for diagnostic purpose. So the secondary antibody, like Dr. Sunita said, mostly the goat is used because we can get large quantities at one sitting.
So the next question is regarding That's another interesting question. Actually, they're asking about any study material or any textbook. Already there's a mention. I mean, Dr. Sunita mentioned about ICAP. This anapattern.org website is very hugely resourceful, I think. But besides that, is there any study material or any textbook kind of thing for this entire topic? That's another question. I think Dr. John, can you?
John sir can answer.
Yeah.
No, actually, what I use here is Euroimmune. I am not promoting any company or such, but Euroimmune, CPC, they have come out with a small handbook. Actually, I have it with me here.
Yeah.
Yes, yes. That's very useful.
That is useful. The ICAP, the other thing is nowadays with what we can do is easy to form WhatsApp groups. That is the advantage of the WhatsApp for academic purposes. You can take photographs and you can send. That's how I keep in touch with my lab also. Yes. Even when I'm away, if they have some doubtful thing, they capture the photo and send it to me, and then we sort it out. So there are books, but they are expensive. If I have been trying to get it, it's difficult.
It's too technical, sir. What I've seen, certain books are too technical to understand. Maybe.
Yeah, University of Birmingham. University of Birmingham has one site.
Okay.
Yeah, okay. But it is a basic site. The ICAP is good, but as I said, sometimes autoantibodies do not occur singly. ICAP deals with single autoantibodies. Multiple autoantibodies, it becomes difficult. So that is my only, what you say, professional grouse against the ICAP.
So the next question is probably this is answered already, but anyway, I mean just
you can share my presentation to you, Dr. Kalita. I will send it by mail to you so that you can share it.
Sure. We'll take it. Actually, I was about to tell that.
No, I can share it. No, not issue.
Absolutely. I am about to take the permission from you. Anyway, so next, this question is regarding this HAP2 and HAP2010. They're asking, I mean, maybe they need some more clarification, like probably they're asking some advantage, disadvantage kind of thing. So, Sunita,
the HEP2 and HEP2010, the only basic difference is more number of dividing cells are present in HEP2010, sir. 10% more mitotic cells are there. So as sir was describing about the mitotic stages, like when you have this HEP2010, what happens is you can beautifully see the cell in the telophase, anaphase. So more number of mitotic patterns you can see. I mean, that helps us in identifying certain patterns, like example, ANA homogeneous, you have DFS70, you have the spindle apparatus, centriole. If you don't have this more number of dividing cells, no, it becomes difficult at times. So that is why this HEP2010 has got an advantage in having 10% more dividing cells, which helps us in, it helps us a lot in identifying such patterns.
Here, I'd like to add, we also test for autoimmune hepatitis.
Yes.
So there, one of the patterns that they give has HEP2 cells, but the screening test they send is ANA, our hepatologist. So we used to find a difference. So then we went for the rat kidney, the other substrate separately, and the ANA substrate, we kept it also for the testing the ANA in the required dilutions. Okay. So the HEP2010 helped us helped us to overcome the discrepancies. Yes. 1 or 2 discrepant results were coming. So that's how, that is the advantage of the 2010. Actually, in practice, it's very useful.
Yes.
Okay. So my query to Dr. Sunita, in the liver mosaic, it's 2010 or HEP2 is used?
Liver mosaic 8, we have HEP2, sir.
Okay. Okay,
We
there's
have
another.
another, we have another profile called, I mean, mosaic called mosaic basic profile 3A, which has got 4 substrate where we have Hep 2010, primate liver, rat kidney, rat stomach. So because we have this rat stomach and rat kidney, we'll be able to see the liver autoantibodies as well. There we have Hep 2010, whereas in liver mosaic 8, we have Hep 2 only.
Okay. Okay. Thank you. So another question for Dr. Sunita. Actually, it's a basic kind of a question. They're asking about this, this antigen you have shown, or this autoantibodies, the targets, like SSA/SSB. They're asking, is this the same one or it's a different one?
So they're
or
different.
What is the meaning of this oblique?
See, SSA, SSB, this is otherwise called as Rho and La.
Yeah.
So technically, if you biochemically, if you ask me, they're more or less the same type of antigen. They specify Sjogren's syndrome. So if you take SSA, there are two types, like Rho 52, Rho 60. Rho 60 is diagnostically significant, whereas Rho 52 is non-specific. Dr. John can add any points to this.
Yeah.
So
SSA, SSB backslash, mostly because the ANA pattern is the same.
Yeah.
Fine speckled. So for that reason, I mean, it's customary for us to put it as SSA backslash SSB. It is more a what you call convention we follow from the lab side.
Yeah. Even NRNPSM, same type of patterns you see. It's a coarse granular. That's why we put a slash there.
So that means for ANA, I mean, this IFA ANA, this is the thing we can use actually. And subsequently, if we confirm it, they become separate, like in blot analyzer. So next, the last question is probably, I mean, it's probably somebody who's a little bit experienced on this. So they're asking about this DFS 70 and what is the clinical implication and difference in male and female. Probably I will give this question to Dr. John actually. So little bit on DFS 70 and clinical implications.
DFS 70 initially was thought to be only in normals. Then subsequently now, I think around 11%, various studies have come, 11%, 15%, they say. It is also associated with autoimmune disorders, SLE, etc., etc. So still, there is no clarity. And another thing is the DFS 70, which is the metaphase plate is changed, like Dr. Sunita showed. That is very clear to distinguish. But when it is, when the nucleoplasm is also well stained, it becomes very difficult to differentiate. So that is one practical point people will face, and it cannot be avoided. So that time we just report it as speckled. Then the rheumatologist or the clinician, especially the rheumatologist, they will, based on the clinical picture, they will decide what has to be done further. So from the lab side, we definitely have this limitation on DFS 70.
Yes, sir.
Because we are reporting a laboratory test only.
Correct. Correct. So there is no question in my side. And Dr. Bania and Dr. Deepika, do you have any queries from your side or from somebody from the participants? Yes, Dr. Deepika or Dr. Bania?
No, sir. No, sir.
No question.
No, sir.
Okay. No question. Thank you so much.
If they have any further questions, they can mail us, sir.
Yes.
Given my mail ID, we can also discuss like that.
Absolutely. So mail ID of yours and Dr. John's, I will share with them if anybody asks, or I will share it anyway. So they can
share
ask,
the phone number also, no problem.
Absolutely. We'll do it.
So
next in the agenda is, we're coming to a close of this workshop actually, and just a small vote of thanks. That is the last part of this workshop. And so here I would like to thank our administration, AIMS Rishikesh, Honorable Director Sir and Dean Academic for allowing this conference plus this workshop. then the organizing chairman as well as the co-chairman. Incidentally, one of the the co-chairman madam is here in this workshop as a delegate, Dr. Neelam Kast. And I would like to thank Professor John and Dr. KG Sunitha for their lectures, their very prompt acceptance immediately without taking any time. They accepted our request. Then organizing secretary, all other participants, and last but not the least, the other co-coordinators here, Dr. Banya, Dr. Chakraborty, and there is Gaurav is also there. And all the technical team, I would like to thank for the kind of help. Without any glitch, actually this workshop was over, and we are very delighted for this. And with that, I would like to request to take a snap. Actually, if all of us can be online with video on, I can see everybody is on actually. Can you take a snap? We'll take it, Dr. Bania or Dr. Chakraborty, or somebody is going to take this.
Yes, sir. Yes, sir.
Is it taken? Okay.
Yes, sir. It's done.
Okay. Thank you so much, sir. Thank you. We will share your presentation with the participants.
Yes,
As
I'll send it down. I'll send it by mail.
Yes, sir. We will get it from you. Thank you. Thank you.
Thank you very much.
Thank you, Dr. Sunil.
Thank you, sir. Thank you, John, sir.
Thank you.
Thank you very much.
Now we can exit.